How you doing, guys? Today we're going to be talking about squirrels and why they get the fur color they do. So we have our two test samples here, previously maimed / killed, whatever you want to call it. So yeah, we have them now.
The color of a squirrel's fur is determined by something called a gene.
You can see here we have our two test examples. This is a fox squirrel. This is a black squirrel. It's black, just like its name implies. So, to start off, we'll be talking about the central dogma of biology. And the first step is transcription.
This happens in the nucleus, and what it does is it makes mRNA from DNA. And what mRNA is, that's messenger RNA. So the first big difference between mRNA and DNA is that mRNA is a single strand, so it's like one helix, whereas the DNA is a double helix, so it kind of looks like it's chasing itself, but it's really not because it's chasing another strand of its nucleotide. So another difference between RNA and DNA is that RNA contains a thing called uracil instead of DNA that contains thymine. It serves essentially the same purpose, it just is a different thing in place of it.
And the type of sugar used is ribose. I bet you didn't know that. Initiation is when the RNA polymerase binds to a location on the DNA known as the promoter. The promoter sequence signals the beginning of a gene.
The enzyme used here is called RNA polymerase, and what this is going to do, this is going to take the DNA template strand, it's going to read it from the 3' end to the 5' end, and it's going to be like, and it's going to tell it what to do.
So, the free-floating RNA nucleotides essentially build an RNA strand from 5' to 3' using the free-floating nucleotide monomers. And eventually, they will get to a point that is called the terminator, or the terminator sequence, as many like to call it. And that essentially tells transcription to stop, and we'll be getting on to our next phase of the central dogma of biology. Alright, so, first of all, we have our RNA polymerase and the terminating point.
This here, we got a nice fancy ribosome right here. It's going to read our RNA strand. Let's go. Let's check this out.
So this is really intense. This is not PG, so cover your eyes if you're under 13.
And then bam, see? It's like this. Told it when to stop.
Yeah.
It's kind of crazy.
It's really frightening for younger viewers.
So with the mRNA strand that's going to be read by the ribosome, not everything is always going to have a purpose. There's a thing called an intron. What that is, it's a set of 3 that does not actually code for anything. It's a sequence that does absolutely nothing for the protein, but then there's the exons, and those are the ones that actually do code for the protein.
Now we have the cap and tail, and the cap allows the ribose to recognize the messenger RNA sequence and bind to the mRNA when translation begins, and the tail is a sequence of 150 or more adenine.
Here's my nice little example of introns and extrons. This would be an example of an exon because it actually means something. But in binary, since only 0 and 1 means anything, this would be an example of an intron because it does not mean anything. It's just gibberish. Ta-da!
Next up, we have translation, which is probably everybody's favorite, but Anyway, location within the cell is in the cytoplasm, and the purpose is to make a protein from the mRNA.
What we got now, we got this ribosome structure. It, believe it or not, is 2 subunits composed up of rRNA and some proteins in there. Then you got these mRNA codons. What these are going to do, it's going to tell them what amino acids to make, and there is actually one that tells it to start. There's not only one that tells it to stop. Crazy, isn't it?
So next up is the tRNA, which is the transfer RNA, and that essentially carries amino acids from the cell that it is in to the ribosome, and then the anticodons, which are composed of 3 base pairs, which complement the codons of the messenger RNA.
We got some amino acids in here. What those are, these are the monomers of the proteins, and they're gonna be linked together chemically to form a larger protein molecule. Then we got these peptide bonds. These are formed between the amino acids, and it's actually a type of dehydration synthesis, believe it or not.
So, there are also proteins that are polymers, and those are known as polypeptides. And then there's also stop codons, as he mentioned previously in the video. There are start codons. What the stop codon essentially does the exact opposite as the start, and it stops amino acids from adding amino acids onto a protein chain. So, yeah. Alright, so this is the start codon for the process.
This is the stop.
The stop codon. This is the tRNA, or transfer RNA. This is the amino acid, which you can barely see, but it's there, see? Right there.
It is there.
Alright, so
Better believe it is.
Go through. tRNA goes through the start codon.
That starts making amino acids.
Yep, so you can see it right there. And it hits the stop codon. Boom.
That's when it stops making amino acids.
Yep.
That's when it fell off. Did you see that?
Yep.
Actually didn't, but it happened. What this protein does, it's going to go back around and it's going to decide what color is our squirrel. And it's going to be like, you know what? I'm having an off day. Let's screw with him and make him purple. And then he's like, Jimmy doesn't have any friends. I wonder why. It's because he's purple.
There is actually a purple squirrel in the world. It was recorded. No scientists know what made it that way, but I'm pretty sure it was the proteins.
But it might have also been a mutation. Tell us a little bit more about this, Brandon.
Mutations?
Yeah.
There are multiple parts to a mutation. There are multiple kinds, I should say. This one is we're going to be talking about a point mutation. And that is essentially when a single nucleotide is changed in an organism. Now the impact on this is pretty clear in that it essentially affects the squirrel fur color protein that we didn't make up at all.
No, not at all.
It affects it, so say that, I don't know, it starts out purple, well maybe it wants to make it a slightly darker shade of purple. Maybe a slightly lighter shade.
So this right here, this is the mythical jackalope, and the wings and the antlers are a result of mutations, believe it or not. And check this out.
Since I have experience with being a mutant, yeah, this one also has wings and antlers, yeah.
Except his antlers are actually party hat.
So that was our interpretation on the central dogma of biology.